Okay. Good morning or good afternoon, everyone. So I'm going to talk about something a little bit different today that you last few weeks in the seminar series. I'm going to be talking about plants. So going back here, just a quick introduction. Again, I'm a faculty at the University of Florida, and I'm also a co-founder of SolusCell. This is a company that was developed to provide single-cell services primarily for plants, but also for other systems. I'll talk a little bit more about this at the end of the talk today. Now, as you see from my slide, I'm going to be focusing part of the talk in what I call plant exceptional traits. I think probably many of you that work with plants, you may be looking and thinking, well, plants do many exceptional things like photosynthesis. They provide us with food and fiber. But the reality is that there are a few certain species that do some truly amazing things. This is just one example of one of these species. This is a species that's native to New Caledonia. And what it does, it actually is able to grow in soil that has very high levels of nickel. Now, the reason why it has sap that is sort of this bluish, greenish color is because it has actually the ability to capture that nickel, take it up to the aerial part of the plant, and it is thought that perhaps it uses it as a way to protect itself against pathogens. Okay, so this is one really amazing trait that this tree has and could have potentially great value for us. For example, if you were to use this species for mining nickel using plants, using plant biology. Now, for you to be able to understand these traits, of course, you have to understand development, right? And development is a field that really began evolving back in the 50s, 60s, and 70s in the last century, mostly with work done in Drosophila, work done in C. elegans, where scientists were for the first time looking at how to manipulate individual genes and see the effect of that manipulation of those genes in, for example, Drosophila development. Now, developmental genetics went through a period of very fast growth back in the last century. And just like any research field, at the beginning, perhaps there's no technology available, then it happens a breakthrough, and then a lot of people jump into this area of research and start studying it. But then very often it goes through a period at which that research field is no longer very interesting, or perhaps something else comes along. And this is really what was happening with developmental genetics back in the 90s and early this century, largely because there was a new field that came along, which was called genomics, right? So everybody wanted to study genomes, wanted to study genes, but really developmental genetics took sort of a back seat. Largely also because genomics was not really providing much new information for developmental genetics until, of course, a new breakthrough happens. And I think we all can be guessing that the breakthrough that I'm talking about here is the development of single-cell genomic technologies, right? So I'm referencing here the paper that this breakthrough of the year by Science in 2018, but the reality is that there's a lot of development that really happened earlier this century in 2010 and so on. But what this really allowed us for the first time is to combine the power of genomics with the ability to understand the development of organisms. And again, I'm just referring here to these two papers, but of course, there's been a whole body of foundational literature that was developed in this area, mostly with animal systems as well as with humans. Now, this sounds great. There's no doubt this is technology that's very widely used, except perhaps if you work with plants. Okay, this is a very simple exercise that I did yesterday. I selected the word single-cell RNA sequencing plus humans or plus plants in PubMed, and I looked at how many publications reference this technology with humans or plants. And you can see that development in this area is vastly, vastly superior in humans than in plants. So we are really at a very, very early stage of developing the technologies, developing this technology for its application to plants. Studies in plants represent perhaps about 2% of studies that have been done in humans. So really a field of research that's at very early stages of development. Now, what's the reason for that? The reason for that is primarily because plants, different from animals, they have what we call cell walls, all right? So every cell is basically in this envelope of lignans, cellulose, and hemicellulose and other plant products, making it really difficult for us to be able to isolate them individually without damaging them, without having RNA leakage. That is one key challenge. The other major challenge is the fact that most of the analysis methods that have been developed to date have really been developed for studies in humans, in animal systems, and not really for plants. So back in 2019, when we became interested in this field and we thought it could be very valuable for the research that we were doing, one of the first steps that we did was to develop the methods by which we could effectively isolate nuclei to use in single-cell analysis, but also to develop the bioinformatics tools that would allow us to apply this to plants. I need to mention several of the people that were involved here. Daniel Conde, who's now a faculty in Madrid, Paolo, who's now a faculty in Italy, Wendell and Kelly, who are both faculty at the University of Florida. They were really critical in establishing both the methods for nuclei isolation and single-cell analysis, but also the bioinformatics tools that were required to apply this kind of technology to plants. Now, one of the first things that we did as we were developing this methodology was, well, can we actually use it to study what we call the shoot apical meristem of a plant? So on the tip of the plant, you have a set of cells that are meristematic cells, or similar to stem cells, that give rise to a number of most of the different organs and tissues in a plant. So we wanted to look at that type of tissue to see if we would be able to recognize all the different cell types that we're expecting to see, and if we could use that to understand how these cells differentiate. This was primarily work from Daniel Conde, again, I mentioned previously, but also Henry Schmidt, a grad student in the lab. So of course, we generate the single cell data, and I'm sure everybody has seen many of these plots before with the different clusters in our UMAP. And here we encountered really the first challenge of doing this in plants, in the sense that for humans and for animals in general, there's typically quite a broad knowledge about genes that are specific to a given cell type. That type of knowledge in plants is very limited, is primarily restricted to Arabidopsis, and very often the transfer of that information from Arabidopsis to other species is very limited. So we spent obviously quite a significant amount of time developing the markers that we needed to annotate these cells, as well as doing molecular work in the lab using promoter GUS and promoter GFP fusions to see where certain genes were expressed and to be able to annotate these cells. Now, one of the things that becomes really evident when you look at this data is how much information we are missing when we're doing bulk RNA sequencing. These are dot plots for a set of different gene families, CCOMT, C3H, and so on. These are genes involved in lignin biosynthesis, genes involved in development. And if we look at just this one gene here, CCOMT, this is a gene involved in lignin biosynthesis, you can see for this specific gene family member here, its expression is very, very specific to the companion cells. If we go back to the previous slide, you see the companion cells are just this very small number of cells over here. So it's pretty evident that if we were looking at the expression of this gene using a bulk strategy, that signal in that specific cell would get diluted across all the cells. And we would probably interpret that information as saying that this gene is either not expressed or expressed at very low levels. So very, very clearly from the start, it became evident that single-cell RNA sequencing would be really critical for us to be able to pinpoint the expression of genes in a much more precise way, and that was going to be actually very, very useful for us. Now, why are we doing single-cell RNA sequencing other than understanding gene expression across different cell types? Another thing that we were really very interested about is understanding how gene expression changes along a cell lineage. And more specifically here in this case, we're trying to understand how cells in the vascular cambium differentiate to form the different cells that are observed in the xylem. Okay, so if you're not familiar with woody plant anatomy, you have a layer of meristematic cells here, which we're going to call, which is the vascular cambium. And that vascular cambium, those cells differentiate either into the xylem in the internal part or into the phloem in the outside part. So could we actually use single-cell technologies to understand how these cells divide and then give rise to the other cell types? So this was data that was generated for about 11,000, almost 12,000 nuclei. Those were nuclei that we isolated from wood, again from poplar, which is a tree species. And of course, based on cell type specific markers that we had identified as specific to a given cluster, we were able to annotate the majority of the clusters that we identified in this data. Again, at the point where we did this study, we had already advanced, and other groups had already advanced quite a lot in identifying different cell types and different markers for them, including in poplar. But what you can see right away is that within this group of cells or nuclei, this group here on the right is primarily representing cells that we see in xylem. The ones that you see here on the upper left, primarily in phloem, and these ones here in the epidermis, endodermis, and the cork cells. Okay, so we can see this very broad group of different cell types represented in this map, which is what we sort of expected. But then you can start actually asking the question, well, what if I now analyze and look at the expression of a given gene that I know is specifically expressed in the vascular cambium, in those cells before they started differentiating, right? So we see one of those genes on the top of the UMAP over here. And then we start looking at genes that are expressed as these cells starting to divide and differentiate into the different components of the vessel, of the xylem. And what we see there is that what we really have is a suite of cells that are gradually differentiating to form all the different cell types that we see in the xylem. So in other words, we are uncovering basically a xylem cell lineage. In other words, the transcriptional program of the cells of the cambium differentiating into the different cells of the xylem. So that's great. You know, this is sort of what we wanted to do. That was one of the goals of this study. But what we also found was a set of cells that follow through this trajectory, but that we really could not tell what was their identity, what was the function of these cells. So what we did here was what you have to do in those cases, you develop a promoter Gus fusion to be able to understand what those cells actually represent. And in this analysis, what we could see is that the cells represented by this cluster over here are primarily those cells that are surrounding vessels. Okay, so again, for those who are not familiar with plant anatomy, you have these very large empty cells. These are the vessels. This is how water is transported from the roots to the upper part of the plant. And these cells that surround the vessels, we call them contact cells. They have a critical role in allowing and helping these cells, the cells from the vessel, respond to conditions of stress. And in the past, we really have not been able to study these cells because they represent very few cells within the xylem, and we really had no way of isolating and analyzing them individually. So this is something that we were able to tell here, that this cluster is representing contact cells. But another question that we had here is, can we actually manipulate this cell lineage in order to perhaps not produce contact cells, but produce other types of cells? So what we did is we identified a gene that appears to be very important, that transition into contact cells. We knock out this gene using CRISPR-Cas, and the consequence of that knockout are plants that have far more vessels. They actually don't seem to have contact cells, and these vessels appear to be much more clustered together. Okay, so we're really looking here at the ability of using the information that's coming from single-cell genomics to manipulate, to change cell lineages to produce, in this case, wood that will have different properties in terms of the different cell types that are represented. All right, now that was one of the things that we were interested about in those early days of single-cell analysis in the lab. The other question that we had was, can we use this information to understand what is different between two different species? Okay, and again, I mentioned to you, we were doing work primarily in poplars, studying the shoot-at meristem, but there was another group that was looking at the same question, but was looking at that question in Arabidopsis. So again, for those unfamiliar with these different plant species, Arabidopsis is a herbaceous plant. It is an annual plant. Poplar is a woody plant and is a perennial plant. So they have vastly different life history traits. So here what we thought was, can we generate single-cell data, or can we use single-cell data that was generated previously for one of these species, combine, integrate that information to then identify perhaps what are the genes that are expressed primarily in one species versus the other along a lineage that could potentially tell us what are the genes that define their developmental differences, right? So this could be very important if you are studying a specific species that has a certain trait of interest, but there's no variation for that trait within that species. You could potentially look for a related species that carries a different trait, a different phenotype, and make a comparison between the two. So we've done that comparison, and here what you see is the trajectory of cells from the vascular cambium, from shoot meristem, differentiating into sieve elements. So each one of the columns here represents cells along this trajectory. And what you have in the rows represent the different cells, the different genes that we characterize both in Arabidopsis and Poplar. And I think what you can see in general, that in Arabidopsis, which is this panel here on the left, and Poplar, the profiles are very similar. It appears that in general, most of their genes are actually quite similarly expressed, both in Arabidopsis and Poplar, as this shoot-at meristem is differentiating into these sieve elements. However, you can also look and use this kind of information to pinpoint genes that are very distinct. And in one case, LAX2 was one of the genes that we were very interested in because we noticed it was very, very highly expressed in the vascular cambium in Poplar, and it had a different pattern of behavior in Arabidopsis. So we downregulated this gene in Poplar, and what we get is actually a xylem, an extent of xylem cells that are much more reduced relative to the wild type, suggesting that this gene could be one of the genes that is part of the developmental differences between Arabidopsis and Poplar. So the intent of these initial studies was really to do a proof of concept, get an understanding of the use of the technology and how useful the technology would be for studying plants, and more specifically for studying Poplar. But then we also thought, okay, can we actually use this type of information now to understand these truly exceptional traits that certain species of plants have, and see what makes them different from other species? And here what I'm going to do is, in the second part, I'm going to focus most of the talk on nitrogen fixation. This is a very important trait for agriculture. But I'm briefly going to mention work that we're doing in photosynthesis, more specifically on chem photosynthesis, as well as in having plants making diamonds. And of course, plants don't make true diamonds, but they make certain plant products that resemble very much diamonds in terms of their properties for recalcitrance. So I'm going to focus mostly on our work in nitrogen, but I'll mention briefly photosynthesis and the work on sporopollenin later. So first of all, nitrogen fixation. This is work that has been led now primarily by Dr. Wendell Pereira, who's a faculty here at the university, who works with us in the lab. And if you're not familiar with nitrogen fixation in plants, let me just say that if you look at a root, most commonly in the legume that contains these structures that look like these little balls, These are likely the structures that are providing a habitat for certain bacteria to live in those nodules. Okay, and what happens is that these bacteria, which I'm going to refer to them as rhizobia here, they can provide the plant with nitrogen, while the plant itself provides these bacteria with carbohydrates. So it's a symbiotic relationship, and it's a very important symbiotic relationship. Now, most plants don't have the ability to form this symbiotic relationship. So for most plants that we use in agriculture, we fertilize them with ammonia or other types of nitrogen fertilizers like you're seeing over here. Now, this process is extremely important for agriculture. It was invented, the process by which you can produce nitrogen fertilizer is very important. It was really discovered by Fritz Haber, who's this guy over here on the left side, and then Carl Bosch is the person who then took that process and was able to set it up in an industrial scale, really creating the ability for us to produce fertilizer today at the scale that we do and that supports today's agriculture. I think very often people don't realize the critical importance of this process, but this is just an estimate of the number of births of the world population. If we did not have the Haber-Bosch process, we would have been around the year 2000, around 3 to 4 million people. But because of the very efficient and highly productive agriculture that was enabled by the Haber-Bosch process, the world population was really able to skyrocket to levels much, much higher, again, in large part thanks to the creation of the Haber-Bosch process. So it's very important, incredibly important for agriculture. There's just one problem, is that for you to be able to capture that nitrogen from the air, fix it in a form that you can provide it to the plant in the form of fertilizers, it means you need very high energy because you need very high temperatures and very high pressure. There are estimates of how much CO2 is emitted because of the Haber-Bosch process, and it's used in agriculture. They vary quite a bit, but numbers are about 2 to 3%, meaning that if we were to be able to replace the Haber-Bosch process by the ability of the plants to fix nitrogen themselves, that would be somewhat equivalent to removing about 17 million cars off the road. Okay, so there's tremendous amount of CO2 produced from the Haber-Bosch process, but the Haber-Bosch process is very important for agriculture, but we would really love to have something that could replace that nitrogen. And of course, one of the ways is by making plants, by giving them the ability to establish that symbiotic relationship and fix nitrogen. Now, one of the great things about this process is that this is not something new. This is something that people have been studying for a long time. And we understand today that for that relationship to happen, the plant needs to recognize the bacteria, needs to recognize the rhizobia. It needs to be able to allow the rhizobia to come in and colonize the root. And most important, perhaps, is the plant needs to be able to recognize all those signals and then create a nodule, which is what you see here in this in the image again. Another advantage of the body of work that had been done in understanding symbiotic nitrogen fixation is that people over the last decade really have gained a very detailed understanding of how the roots of a nodulating species, how do the cells of those roots differentiate to go from a state that you would see in a normal root to a root that contains a nodule. Okay, so in other words, we know very well, for example, that the layer of cortex cells, layer 4 and 5 in Medicago, will differentiate to form this fixation zone in the nodule. Okay, and this is, again, something that we view as very critical before we start any single cell study in a plant species for any trait. We need to be able to, we like to try to be able to understand as best as possible what is the cellular program that is involved in the differentiation of those cells into a new tissue, a new cell type, or a new organ. Okay, so that's very, very critical. Another thing that we learned when we were performing this study, and we realized was really quite critical at the time, was how important is for you to work with a given tissue where you have a large population of cells that is responding to the stimulus that you want to study. Okay, this UMAP here on the right. This was a study that was published a couple of years before. We completed our part of the work. They were looking at the same species and also Medicago truncatula and roots that were treated with rhizobia also just the same way. But what you can see in this data that was produced 48 hours after the inoculation is that you have very, very few cells that seem to be responding to the inoculation, okay? And I think this is really a reflection of the fact that when you have that process ongoing, very, very few cells on the root actually do respond to the rhizobia, and therefore you also see very few cells in your single cell analysis that will display a unique pattern that you want to study, okay? So thankfully, when we started this work, we began first looking at single cell data in a wild type. This is called genotype A17. But we had a collaborator in Clemson in South Carolina who approached us and she said, listen, why don't we, instead of just looking at the wild type, also look at this mutant that we call the SUN4 mutant, okay? And what this mutant has, it lacks the ability to control the number of nodules. In other words, the roots of this mutant produce a very large abundance of nodules. In other words, it makes it very suitable for us to study using single cell analysis. So we generated single cell data for the wild type, we generated for the mutant, we generated 0, 24, 48, and 96 hours. And here's the data that integrates both the data from the wild type and the mutant across all the different cell types. And I think it's pretty obvious as you look at these images that as you approach 48 and 96 hours, you have the emergence of this whole set of new cells or new types of cells over here, right? So of course, the first question is, well, what are those cells? What is really emerging in this UMAP at 48 and 96 hours? And of course, what you do then, you look for specific markers, specific genes that are specifically expressed in this, what I'm going to call here, cluster 6. And what we notice that obviously these are all markers for the nodule. Okay, so obviously the set of cells that is emerging, that is forming this cluster 6, represents cells from the nodule that are appearing after 48 to 96 hours of treatment with the rhizobia. All right, very good. But this is nice to characterize expression across these different cell types. But what we really wanted to understand is how can we use this information now to see how cells from the cortex of the cell of the root will differentiate into the nodule meristem and the different parts and all the different parts of a nodule. And as I mentioned previously, we were lucky to have really a pretty significant body of work that had been done before that gave us the information that we needed to better understand differentiation of those cells. So what did we do for this? Well, we took the cells that we had identified as representing cells from the root cortex, as well as those that were representing the nodule. And what we did is we retrieved those cells and we reanalyzed these cells by reclustering them. And this is what you see here on the right side of the panel. And when we analyze these sets of cells, in other words, the cortex cells differentiating into the nodule, what we could see through trajectory analysis that we have basically two different trajectories. We go here from cluster 2, 1, 4, 7, and then when we get to 3, that cluster splits into two separate clusters, cluster 6 and cluster 8. In other words, we have two separate lineages that are emerging here. So what we did is then we annotate what these clusters 6 and 8 represented. We were lucky to have single cell. We had actually laser microdissection data that had been generated previously, so we could tell that this cluster 6 likely represents mostly cells from the nodule and infection zone. and these ones represent cells from the nodule in the meristem, so that what we really have is a trajectory going from nodule meristem going from the nodule primordium into the meristem and into the infection zone. And again, one of the things you can look at based on that trajectory is to look at how specific genes or classes of genes that you know are important in the process are expressed. Just a little bit, looking into a little bit more detail, just to finalize the description of this work. We looked into a little bit more detail how these cells from cluster 3 differentiate into cluster 6. These are some of the genes that we identified as differentially regulated. You can see them along the trajectory. And some of these genes were genes that had been considered as candidates to be involved in the process, but had not really been studied. Stylish 4 is one of those examples where it has very little expression in the cortex. That expression really shoots up as we get towards the formation of the nodule. And what we saw is that when we do RNAi interference on this gene, indeed, we obtain plants that have far fewer nodules, okay? So clearly, we see single-cell genomics as a very powerful approach to understand the development of processes like, for example, nodule formation, but other processes as well. I'm gonna switch gears and talk very briefly now about some additional work that we're doing in this area. One of them is work in chem photosynthesis. This is work that is led by Noé Perron, who's a PhD student in the lab. And what Noé has been interested about is chem photosynthesis. So just for those that are not familiar with C3 and chem photosynthesis, most plants that are used in agriculture, they do what we call C3 photosynthesis, meaning they open their stomata during the day when CO2 can be captured and then converted into sugars. But in that process, also, you have a significant amount of loss of water through evapotranspiration. Now, during the night, stomata closes, you don't have any photosynthesis happening, and you also don't have any water loss, but that doesn't really matter because at night, water loss is very limited. What these plants that perform chem photosynthesis do that is very unique is that they have the ability to open their stomata at night to capture that CO2. Then that CO2 will get stored in the form of malate to be converted into sugars in the next day. In the next day, during the day, the stomata is closed, so there is no water loss. So these chem plants, they can achieve 3 to 6 times more water efficiency, use efficiency than C4 and C3 plants. And as you can imagine, it would be extremely valuable to be able to introduce this into agricultural crops. So we know a good deal of information about the different pathways that are involved in this. I'm not going to go into this in detail, but what Noé did was to take a plant that we know is able to do both C3 photosynthesis and chem. It does chem when you do not provide, when you provide some sources of abiotic stress like drought or salt treatment. He applied a treatment to some of the plants and collected those plants leaves from those plants after 8 days. So we have both plants that are doing C3 after the treatment, but also plants that were previously doing C3 but are now doing chem. So obviously, single-cell analysis is a wonderful tool for us to use that information and be able to perhaps identify what makes this, what is necessary for triggering chem photosynthesis. This is the UMAP plot that was generated for this data. Just very briefly, again, we have a set of cells. There are mesophyll cells where we know photosynthesis, chem and C3 happens. These are these clusters over here. But perhaps most importantly, within these mesophyll cells, we were able to tell precisely which ones are the ones that are performing both C3 and chem at dawn, but also cells that are performing specifically C3 or chem at dusk. Okay, so for dawn, we really did not see a significant difference between cell expression in C3 and plants doing C3 or chem, but at dusk, there was a drastic difference. So The approach that we usually take for this kind of data, for this kind of study, is that we apply a network analysis. We have been used high dimension WGCNA quite extensively. It's been very powerful for us. And in this approach, using this approach, you can identify, we've identified this module 2 that has a very high prevalence of genes that were responding to the treatment, as well as genes that are expressed specifically in those cells of the mesophyll that are performing either C3 or CAM. Again, I'm not going to go into a whole lot of detail of the study. There's a lot of it is currently in bioRxiv. You can take a look at that publication for more details. But what's perhaps most important is that this type of data is allowing us to now elucidate what are the transcription factors that are critical to trigger the beginning of CAM, to trigger that differential regulation of stomatal closure and stomatal opening, and also recognizing the calcium signal that happens when you have a condition of drought. Okay, so this is again the second type of trait that we've been very interested. And finally, the last one, I'm just gonna brush very quickly through this. Plant diamonds, of course, plants don't make diamonds, but plants make a plant product that's called protopolynin that is very similar to diamonds in terms of its recalcitrance, in terms of its the challenge to destroy it. Okay, it's a very, very rigid material. And of course, that is the material that coats pollen grains. Now, we have received funding from the Department of Energy or SHOT program with the idea that if we could introduce some sporopollenin in the roots of plants, that could create a potentially near permanent storage of carbon in the soil. So obviously for carbon sequestration, it's very important. And from our calculations, we see that if we engineer the 36 million hectare US maize crop so that these plants had 5% sporopollenin in the roots, we could sequester up to 54 million metric tons of CO2 per year. Okay, so obviously engineering this would be very important. The problem is that sporopollenin is produced very specifically in the tapetum cells in the anthers. It's not produced in the roots. So what we have done is we've taken buds and we've taken anthers from poplar and we've characterized them using single cell analysis. There is some understanding of the enzymes that are involved in the synthesis of sporopollenin, but there's very little understanding of what are the transcription factors that control that. And again, I'm just going to go very quickly through this. The single cell analysis of anthers allowed us to identify a couple of clusters that represent tapetum cells. And in that process, again, using WGCNA, we could identify a very specific transcription factor that's expressed in that stage where sporopollenin is being produced. That is a transcription factor that we're going to be manipulating, introducing in roots to see if we can induce synthesis of sporopollenin in that organ. So stay tuned. This is really work that is in the first year of this project. And I think we should have some data on this, more data on this very soon. So to summarize, we're really entering a new era of plant biology where understanding development is now no longer really a privilege of those that work with model species. Most of the studies that I've shown you are really at early stages of development. There are very few publications. So there are tremendous opportunities for growth in this area. And virtually, I think every trait can be studied using these strategies, from quantitative traits, including growth, as well as traits related to flavor, ripening, and more complex developmental properties. And most important, I think, is that now identifying the genes that are implicated in differences in development between species is much more straightforward than it was before the development of this technology. Now, I mentioned to you that plants are really lagging behind in terms of development in this area, with very, very little work done on this. And that is part of the reason why, that was part of the rationale for us to create SolusCell. SolusCell is a company that was founded about a year ago, where we took all that knowledge that we had developed to isolate nuclei and do single cell analysis, but we now apply that to any client, anybody that is interested in performing those analyses in their own materials. So in other words, we go from tissue, doing nuclei isolation, very often from difficult to dissociate tissues, to single cells, and then we do the data analysis. And again, this is an effort by a team that involves Christopher Vinnes, who is the director of optimization, Mariana, who directs all the operations, and Wendell, all the data serves. You can look more about SolusCell in our company website. A little bit about the services. We can just create libraries or create libraries and do the sequencing. But perhaps what's really critical, what's really important for this talk, since we're talking about Singleron, is that when we developed the company, we knew very well how we could isolate nuclei efficiently for plants, for a broad range of different tissues. And we knew how to analyze the data. But the big question here was, well, what is the platform that we're going to use to actually generate the single cell data from those nuclei? And after we sampled, we tested a number of different platforms. We converged towards the decision to use the Singleron platform as the way to generate information, as the platform that we prefer for generation of single cell data by SolusCell. So this is what I wanted to bring in this talk today. Again, some of the key individuals that were very important in generating the knowledge that I presented in this talk, as well as also Noé, Ellie, and Hank, who are grad students working and generating some of the data that I talked about today, collaborators in Europe and Canada, as well as the University of Wisconsin and South Carolina. And most of this work has been primarily funded by the Department of Energy, either the gene function program or also the biosystems design program, as well as this new Earthshot for Energy program that was launched recently. And with that, I can take any questions that you may have.
Thank you. Thank you very much. That's very, very interesting. And I had one question. So you mentioned this point twice about the lack of single cell sequencing data or the dearth of data compared to human in plants. Why do you think that is? Is it lack of researchers, lack of interest, or is it the difficulty?
So I think in general, if you think about what is enabled by single cell genomics, which is, one of the greatest things is development, is understanding development. I think the potential actually in plants is far greater than in other organisms because there's tremendous variation in terms of development. I think the two points that I mentioned at the beginning of the talk, the challenge in being able to isolate high quality nuclei or high quality cells is truly a barrier for most scientists. Most research labs are not geared towards the development of these protocols. And the other fact is that single cell, the annotation and the analysis of this data can be somewhat, to some extent, different than what is used for animals. So some of that technology, some of those approaches need to be changed to be adapted to be used in plants. I think those are the main issues.
Okay. Okay, so we have some questions here. First question, I'll read it out. Is there any effort of generating genome data for different forest trees for single cell research, for example, beech tree? And if so, what are some of the examples?
Yeah, so, I think surprisingly, if you look at the work that has been done with single cell genomics in plants, you'll find that many of the studies that have been published have been in Arabidopsis roots. That has been a model that has been studied for a long time, but actually also in wood development. I don't think in beech tree, but populus has been a species has been extensively studied. Again, I showed a couple of studies early in the talk, but there have been groups in Taiwan, China, as well as in Scandinavia that have done single cell analysis of trees. Again, I don't think the genome sequence today is a significant barrier because we can generate it at a relatively low cost. So the work that I mentioned for for chem photosynthesis, this is in the common ice plant. I don't think I even mentioned the species there, but it was a species that when we started the study, there was no genome available. So we had to generate the genome sequence and the annotation a priori to the single cell data. Now, about other trees, I mean, I'm aware that of studies using single cell analysis of conifers like pinus, work that has been done or is being done potentially in eucalyptus. So there are definitely some other tree species that people have been interested in studying using this kind of strategy.
Another person asked, did SolusCell collaborate with studies involving plant pathogen versus host gene-to-gene interaction in single-cell level?
Yeah, so I cannot go into details of course of studies that we do within SolusCell, but I can say that within the work that we do at the university, we have collaborated with other scientists that are interested in understanding the interaction between a given pathogen and the plant tissue, right? I think if you take a given plant tissue and you apply a certain pathogen, what you will get if you do bulk RNA sequencing is the response of those cells across different stages of the pathogen infection. One of the advantages of single cell is that when we do this kind of analysis, we can actually separate cells at all the different stages of that pathogen infection and understand how the plant is responding to the pathogen at all those different stages. So it can be very, very powerful. And it's something that we have done here as part of collaborations here within the University of Florida. But of course, there are many people out there that are interested in using this approach because it could provide a lot of answers about the interaction between plant and pathogen.
Thank you. So another question, and yeah, this is an observation. So someone noticed that all your work focused on the nuclei. What are your thoughts on using protoplasts in single-cell experiments?
So that's a very good question, and I'm sure it comes up. It comes up a lot. When we started this work in Pauper, again, these were the early studies that we've done back in 2019, 2020. What we notice is those treatments to generate protoplast can be quite inconsistent depending on the type of material we're working with. We were interested in a shoot at comeristem. We were interested in the differentiating xylem, which means we have layers of cells that are highly lignified. They're very difficult to separate, while other layers, for example, the vascular cambium has very little lignin. In other words, those cells can be very easily separated. The consequence of that is that as you apply a treatment, an enzyme digestion to separate the cells and generate the protoplast, you get a lot of differentiation in terms of the representation of those cells. Now, we have a study that is being concluded, is probably going to be submitted in the next 1 or 2 months, where we took the data that we generated using nuclei and we compared that to the data that was generated using protoplast in the developing xylem in Pauper. So all those studies that were done before with the one that we've done now that I talked about. What we noticed was a very high consistency for most of the clusters, for most of the cell types, but certain cell types were clearly overrepresented or more highly represented in our data, and some were more highly represented in the protoplast data. What was more highly represented in our data were those cells or those nuclei that are more embedded within the differentiating xylem. So for example, those contact cells that I talked about, they're highly represented in our data set, but in the protoplast data set, they're almost absent, again, because the digestion and separation of those cells is very limited. What we did see is also that in the nuclei data, we had a lower representation of cells undergoing programmed cell death. In programmed cell death, one of the first things that gets degraded is the nuclear membrane. And therefore, if those cells are going through cell death, that nuclear membrane will get degraded. And of course, we're not going to capture it in a nuclei approach. But in general, that's one observation that we've done in this comparative study. Many of the tissues and organs that we work with, it would be simply impossible to generate protoplasts from them without having a very different representation of different cell types, or just without having extensive RNA leakage and degradation.
Thank you. That's quite an elaborate answer. I have, maybe this will be the last question. I think we're running out of time. But about symbiosis. So there are these genes that were upregulated, I guess, in the plants where you had more nodules. Do we know the function of these genes? Do they build tolerance?
So when the root is infected by the rhizobia, it needs to be able to allow that the bacteria to come in. In other words, it needs to actually downregulate a lot of genes that are involved in defense. So sort of surprisingly, what the response very often is not a response of trying to create a defense against that symbiotic bacteria, but rather actually to downregulate those defenses so that symbiotic bacteria can get in and can colonize the root and then colonize the nodule. So it's a completely messed up way to respond to a bacteria compared to a normal pathogen. But that's again one of the things that we're trying to understand is how does that response happen so that potentially you could introduce that in crops that don't fix nitrogen. Okay.
Thanks. Yeah, I would like to thank the speaker. I would like to thank all the speakers from the series because it was quite an educational series. I learned a lot about single-cell sequencing in different species. And yeah, so
thank you. Bye-bye.
Bye.